Introduce yourself in 1 minute.
Good afternoon, everyone. Myself, Shomi Pal Chaudhary. I'm Assistant Professor and HOD, Department of Nutrition, Mushadal Girls College, and I am pursuing my research from Calcutta University.
What is that paper?
This is a paper on nutraceuticals and gene expression.
What is that piece
it.
of paper?
No, sir.
Do you think that
No, sir. This is a.
during a competition and delivering a lecture, you can keep some cheats with you?
No, sir. This
Please
is my fault.
submit it to me.
Oh, no, sir. Yes, sir.
You are lucky enough that I have not asked you to step down immediately. If teachers use such pieces of paper, then what will the students learn? Start your lecture, and it has to be completed within 14 minutes.
Today, my topic is nutraceutical and gene expression, unlocking the genetic potential for health and wellness. It's not working. Please help, it's not working.
It is not moving.
No.
You will start fresh when downward start.
It's not working, sir.
চলছে না পয়েন্টার? ব্যাটারি নেই নাকি?
না ব্যাটারি আছে। আচ্ছা ঠিক আছে, ওকে।
ঠিক হয়েছে? সুমনকে বলুন এইখানে তার ডিউটি।
আচ্ছা ঠিক আছে।
আমি তো ডিরেক্টরকে জানিয়ে দেবো যে ডিউটিটা এইখানে।
ইন্ডিকেশন আই will check.
Okay. So I'm starting, sir. Yeah. My topic is nutraceutical and gene expression, unlocking the genetic potential for health and wellness. Next slide. Okay. First, this is the introduction. What is gene? Gene are made of sequences of DNA, which is made of nucleotide, and genes are the units of heredity that contain instructions of an organism's physical and biological traits. So, genes are regulated by complex array of responsible elements that influence the rate of transcription. Now, nutrition and nutrients, hormones, either act directly to influence these rates or act indirectly through specialized signaling pathways. Next slide, sir. So, now the term is nutrigenomics. What is nutrigenomics? Nutrigenomics is the study that study that how gene and nutrients are working together to influence our health. So it's establishing the effects of ingested nutrients and other food components on gene expression and gene regulation. Now it focuses on how individual genetic variations affect the way people metabolize nutrients and how specific foods or diets can influence gene expression. Now the term is very associated with these two words, nutrigenomics and nutrigenetics, that is nutraceuticals. This term is coined from two words, that is nutrition and pharmaceuticals. So nutraceuticals can be defined as a food or part of food or nutrient that provides health benefits, including the prevention and treatment of a disease. Next slide. Next slide. So now genetic polymorphism. This is a very related term with this nutrigenomics. Genetic polymorphism can be defined as the occurrence together in the same population two or more than two alleles such that the frequency or rear allele is always 1%. The variation in DNA sequence can lead to the difference in traits among individuals, including physical characteristics, susceptibility to disease and responses to drug. So, the example of genetic polymorphism is single nucleotide polymorphism and insertion deletion polymorphism. Next slide. Here I am showing a genetic polymorphism schematic diagram. Actually, genetic susceptibility polymorphism mainly caused by gene and nutrient imbalance. Next slide. Nutrigenomics is a systematic biological approach. It happens in the molecular level mechanism, thus providing various type of disease prevention and it has a cause and effect relationship, dynamic cause and effect relationship between nutrition and human genome. For that, nutrigenomics and nutrigenetics are working together. Next slide. So how nutrition interacts with genetic polymorphism? Epigenetic modifications, regulates of gene expression, influence of disease risk. And between these, epigenetic modifications mainly done. Certain nutrients can modify gene expression without altering DNA sequences. For example, folate, vitamin B12, choline are involved in DNA methylation, which regulates gene activity. Next slide. So the main aim of the review is to recognize the specific nutraceuticals which can interact with the genetic code of all nucleated cells and influencing the health outcomes. Next. This is the pathway of dietary signals, nutrients. Nutritional factors can influence the gene proliferation, which refers to the regulation of cell growth, division and replication, and these occurs through various mechanisms such as epigenetic modifications, transcriptional regulation, metabolic signaling pathway. Next. This is the diagrammatic one, that's the nutritional effects on gene proliferation, folate and vitamin B12. DNA methylation, by DNA methylation and synthesis, it effect on gene proliferation, essential for proper DNA replication and cell division deficiency, etc. Such as omega-3 fatty acid, vitamin D, etc. Next slide. Polyphenols, antioxidant and anti-inflammatory action, inhibit tumor growth by modulating gene expression in cancer cells, such as glucose and high glycemic index foods. Excess glucose can overstimulate insulin-like growth factor, increasing cell proliferation, cancer risk. Next slide. This is a schematic diagram how health effects of nutrients and non-nutrients of food relates specific molecular interaction by transcriptional control, transitional control, post-translational modification, etc. By food, nutrients and its outcome ultimately affect our health. Next slide. This is a diagram that how bioactive food components influence genetic and epigenetic events that regulate the different hormones, help to differentiate cell, DNA repairment, cell growth, apoptosis, carcinogenic metabolism, and etc. Next slide. This is a pathway, dietary factors regulate the methylation of DNA, and finally it maintains the cell homeostasis. Next slide. Here I am showing the nutraceutical classification. First one is nutrients, substance which establish nutritional functions such as vitamin, minerals, fatty acids, amino acids, etc. Next one is herbs, phytochemicals. And third one is dietary supplement, probiotics, prebiotics, etc. And nutraceuticals also classify as potential and established nutraceuticals. Next slide. Nutraceuticals which have possible influences on genes. So refined carbohydrates, trans fatty acid, excess saturated fat, excess linoleic acid, all have adverse effect on the genes, whereas monosaturated calcium, magnesium, potassium, iron, zinc all have beneficial influence on our genes. Next slide. This is a continuation of the first one. Vitamin A has beneficial effect, pyridoxine, thiamine, riboflavin all have beneficial effects on our genes. Next slide. This is the health benefits of nutraceuticals. Beta carotene, from which we get from carrots, various foods. This neutralizes free radicals which may damage cell. It protects from cellular damage which caused by different type of free radical reaction. Lycopene, from which we get from tomato. Flavonoids also neutralize free radicals which may damage cells. Next slide. This is the continuation of the previous slide. The soya protein which we get from soybean and soya-based products. Lactobacillus and bifidobacter that act as probiotics, and oligofructose and inulin that acts as prebiotics which we get from yogurt, curd and other dairy product, improve our intestinal gut health and systemic immunity. Next slide. Next slide. Now phytochemicals. Next slide. So phytochemicals acts as a very good nutraceuticals in our daily life. Tocotrienols, tocopherols which we get from grains that suppress the growth of diverse tumor cell lines by initiation of apoptosis Apoptosis is the programmed cell death. An arrest of cells in G1 phase of cell cycle. Carotenoids, fruits and we get from fruit and vegetables. It acts as a very good antioxidant, protect against uterine, prostate, lung cancer, digestive tract cancer, leishmaniasis we get from citrus fruit, inhibiting phase 1 enzyme and inducing phase 2 detoxification enzyme in liver and provide protection against lung tissues. Next slide. This is the continuation of the previous slide. Next slide. So, health benefit areas covered by nutraceuticals, anti-arthritic, prevent rheumatoid arthritis, sleeping disorder, digestion problem, prevention of cancer and growth of mutagenic cell, osteoporosis, blood pressure, cholesterol lowering, diabetes, etc. Next slide. So, nutrients, nutraceutical and gene, the prevalence and mortality associated with multifactorial polygenic diseases such as we are suffering from today, that is hypertension, coronary heart disease, diabetes, cancer, are influenced by a combination of genetic susceptibility and environmental factor. So, consumption of refined food, mixed meals further activate NF-κB driven by free radical production in mononuclear cell. And nutraceuticals, antioxidant, micronutrients, vitamin, minerals, coenzyme Q10, omega-3 fatty acids all have potential to mitigate this type of inflammatory effect and cancerous effect. Next slide. This is the continuation. So, it is known that genes are very important in determining enzyme receptors, cofactors, structural components involved in regulation of blood pressure, different type of metabolism, coagulation factor. It seems that these phenotypic expression may be silent by targeting simple sequence differences known as single nucleotide polymorphism by nutraceuticals and slowly absorb wild food rich in micronutrients and antioxidants. Next slide. So, nutrigenomics, including epigenetic modification, is a rapidly evolving field with the potential to transform public health and personalized medicine by integrating genetic insights into nutritional strategy. Next slide. This is a reference from I get this data. Next slide. Thank you.
I'm really sorry to say, this is a competition for Young Scientist Awards. A scientist is supposed to carry out some research. Where is your research in your presentation?
Actually, sir,
I am not
I've submitted for paper presentation, sir.
indicator of any essay competition.
No, that's fine. She's presented, sir. That's okay.
No.
No, sir. Actually, I've just submitted the paper to present it. So, I don't know that it is a competition or not. I don't know that.
Scientist award, scientist means one who
No, sir.
does something.
When I come here, then I came to know that my name is here. So,
Anyway, I think, in my opinion, you have to improve yourself
yeah,
in future.
it's not my own work, sir. It's an overview work.
But that's fine. This is a wonderful, this is a good idea.
It's a good idea, but not suitable for Young Scientist Award competition. Anyway, so good luck
That's
for
fine.
the next time.
She's presented. That is good.
That's
Thank you.
Thank you, sir.
Thank
Thank you, sir.
next speaker. Get ready. Wait, please. Next speaker will be Dr. Kuntal Ghosh. Wait please, 1 minute. I repeat the rules and regulations for him once again. Mr. Speaker, wait please. If one has to complete his talk within 14 minutes, hardly 1 more minute can be given. 15 minutes is really late. Not more than 3 questions will be allowed from the audience. Preferably the questions should be from the students or researchers. rather than the teachers, to avoid all kinds of conflicts of interest. So you can, you have to introduce yourself very briefly. But before that, whether as a young scientist, contender for young scientist, if our organizers will be felicitating him or not.
No, it's no need of the felicitation or anything. Okay, I am Dr. Kuntal Ghosh. I am presently working as a vice principal of this college. So,
Busy person.
no, it's not like that, sir. It's okay. So today I am going to present a research work, basically that entitled as Diversity of the Bacillus subtilis infecting bacteriophages in the Korean soybean-based fermented foods and its impact in the soybean fermentation. So we know that the indigenous fermented food is one of the oldest biotechnical method for preservation of the fermented food or preservation of the substrate specifically. Basically the microorganisms, mold, yeast, lactic acid bacteria and bacillus, they ferment the substrate, specifically the locally available food substrate organoleptically and biochemically, and produce the different kind of the nutritious product. So next, the soybean based fermented food. Actually, in West Bengal, there is no such soybean based fermented food. Even in the India, you can see the soybean based fermented food only in the hilly region, maybe the Sikkim or some part of the Arunachal Pradesh. Not much more is popular. But in South Korea, these soybean based fermented foods are very much popular. People use as like our the pickle, as like যেমন আমরা প্রতিদিন pickle খাই. So the people in the South Korea, they are using the soybean based fermented food in their daily meal. So if we see the preparation process, you can see the basically there are Chonggukjang, Kanjang, Doenjang and the Gochujang. These are the specifically the fermented foods names, soybean based fermented foods names. So what they do in its preparation, these are the specifically cooked soybean. They ferment it for 2 to 3 days with the Bacillus subtilis, which is a gram-positive bacteria and the major fermenting microorganism for the soybean based fermented food preparation. And then this Chonggukjang is prepared. This preparation process is very short, only 2 to 3 days we need. On the other hand, you can see there is another 3 types of the food. One is the Kanjang, Doenjang and Gochujang. For that, we need to prepare this meju. Meju, what is that meju? Meju specifically, the rice straw is used to wrap the cooked soybean and allow to ferment it for 20 to 90 days. So in between that time, the microorganism present in the rice straw, specifically the Bacillus subtilis, they ferment it and produce this kind of this structure. This is called this meju. So now what they do, this meju is just deep into the brine salt solution and allow to ferment again for 15 to 20 days. And after that, the liquid portion is specifically called the kanjang, which is the soy sauce. And on the other hand, the solid part is called the doenjang, which is similar to the Japanese miso. And another one fermented product is there. This is the gochujang. So the rice powder, salt and the red pepper is mixed with this meju and then again allow to ferment for some times. Then it is called the gochujang. So why this type of the fermented foods are popular there? Basically, this type of the fermented foods, you know, the taste is very good, as like our pickle. So that's why they are using. On the other hand, you can see there are numerous health benefits effect. There are numerous research on those products as they are the indigenous food product of the South Korea. So that's why they have a lot of research on that. So you can see there is this type of fermented food have anti-cancer activity, anti-obesity, anti-hypertensive activity, and also they have the antioxidant activity. So now the problem is the bacteriophage. Bacteriophages are specifically ubiquitous in the nature. They usually kill the bacteria. In the morning, I think we heard the lecture of Professor Devprasad Chattopadhyay. He mentioned about the good effect or the beneficial effect of the bacteriophages because it can kill the antimicrobial resistance bacteria. But for here, for the industry people or the fermented food fermenting company people, this is the nightmare. These bacteriophages are a nightmare to them, as because they kill the fermenting bacteria. That's why they delay the fermentation process and also yield the low quality product, and eventually it leads to the financial losses. So these bacteriophages, specifically they have two life cycles. We all know that. One is the lytic life cycle, another one is the lysogenic life cycle. So in the lytic life cycle, the bacteriophage's DNA is injected into the bacterial body, and after that this bacteriophage's DNA multiplied it or produce its progeny and then lyse the bacterial cell or kill the bacterial cell, and they come out to the environment. This is the lytic process. For the lysogenic process, basically the bacteriophage's DNA is integrated with the bacterial DNA. This is the lysogenic process. So for the industry people or the fermented food industry company, these lytic bacteriophages are the main problem because they can kill the fermenting microorganism. So considering that, okay, it's not like that. Actually, there are a lot of research on the dairy-based fermented food where the lactic acid bacterial phages hamper the fermentation process. But still, there is a very few research on the soybean-based fermented food or its associated microorganism. So that's why we aim to check the diversity of the Bacillus subtilis infecting bacteriophages in the soybean-based fermented food and its impact on the fermentation. And we set the three objectives. First of all, to evaluate the incidence and contamination level of the Bacillus subtilis infecting phages in the Korean soybean-based fermented food, to establish the importance of the Bacillus subtilis infecting phages in the Cheonggukjeong preparation, which is very easy to prepare, and this Cheonggukjeong is similar to the Japanese natto. And again, the third one is the whole genome sequence analysis of the Bacillus subtilis infecting bacteriophages and find out the signature gene for their early diagnosis. So first of all, this is the prevalence of the Bacillus subtilis infecting bacteriophages. So we collect 50 different types of the soybean-based fermented food from the local market, and we check their incidence rate of the Bacillus subtilis phages. So we found there are around 78% of the samples are contaminated with the Bacillus subtilis phages. So its number is really huge. So then what we check, what is the contamination level? Here you can see the contamination level is as low as 6 and it is the 10 to the power 4 level. So the contamination level of the Bacillus subtilis infecting phages are also very high. So definitely they hamper the fermentation process or they delay the fermentation process. So then again we check the rice straw, which is basically used for the meju preparation. We found that the 60% of the samples contain the 60% of the rice straw samples contain the Bacillus subtilis infecting phages. But we couldn't find the exact number of the Bacillus subtilis phages for the two samples. We found only for the one sample, which is around 18. So then we isolated it, 18 different types of the Bacillus subtilis infecting bacteriophages from those fermented food. And here you can see, I think this is very, it's very tiny, we cannot see it properly. But here you can see, this is the BSP7 phage, which has infectivity rate of 77.8. So what we did, we checked whether this bacteriophage can infect any other type of the strains of the Bacillus subtilis or not. Because for the industry people, what they do, if the fermenting microorganism is susceptible to the bacteriophages, then they change the strain. If they change the strain, that time the bacteriophage may not work on that strain. So that's why their fermentation process can go on without any delay. But here we found that the 77.8% infectivity of the BSP7. So definitely if they change the strain also, Bacillus subtilis strain also, that hamper the bacteria, others bacterial strain also. So that's why we cannot say is that the repeated culture or the rotating the fermenting microorganism, it's not the solution for this bacteriophage infection. So this is the transmission electron micrograph picture. Here we found that the 15 phages, those are specifically the myovirus-like phages, and this is the cipovirus-like phages. We found 2, and 1 is the podovirus-like phages. This is the tiny microorganisms.
What is the measurement of the scale used?
Okay, the scales are basically 50 nanometer for each cases. Okay, it is not written, I am sorry. Then we checked what is the effect of these phages in the fermented food preparation. Specifically the Cheonggukjeong preparation, we use this as it is very easy to prepare. So here we prepared the 4 different sets. One is the control and another is the phase treated. So for all of these sets contain the soybeans, cooked soybeans with the Bacillus subtilis there, and for this control sample there is no bacteriophage and for the all of the other 3s, here is the 10 to the power 2 PFU per gram, this is the 10 to the power 4 PFU per gram, and this is the 10 to the power 6 PFU per gram of the bacteriophages. PFU means the plaque forming unit of the bacteriophages we added. Here you see in the control samples after 72 hours or after 24 hours, you can see there is something whitish material is present. This is specifically the polydigamma glutamic acid or the gamma PGA, because the Bacillus subtilis produce this kind of the sticky substance. This is specifically sticky substance which has numerous health benefits. But you can see in the bacteriophage samples there is no such kind of the things. So there may be two possible reasons. One is the bacterial growth is very less in this bacteriophage treated samples, that is likely, and we can see it that this is the control sample, the bacterial growth number is around 10 to the power 9 and it is gradually decreased when the bacteriophage numbers are increased. Even after 72 hours, bacterial number though increased, but we couldn't find any kind of this whitish type of the material or the gamma PGA. So next we checked what is the actual, another one possibility is that this the gamma PGA might be degraded in the bacteriophage treated sample. So that's why what we did, we collect the gamma PGA from all of these conical fluxes by the ethanol precipitation, and then we run it into the agarose gel electrophoresis. Basically this gamma PGA is around 2000 kilodalton molecular weight, so we cannot do it in the polyacrylamide gel electrophoresis. This one is the control sample. Here you can see it's a completely intact, and these 2, 3, 4 are the phase treated. You can see they are the degraded material. So it is clear that this bacteriophage might have some role on this gamma PGA degradation. So to check whether this bacteriophage is really degrading this gamma PGA or not, we did another experiment. We did the enzyme activity, gamma PGA hydrolase activity. So this one is the standard gamma PGA. This is the molecular weight is around 750 kilodalton, and this one, this 6 lane, we treated this gamma PGA with the distilled water and allowed to incubate for 30 minutes. This 7 gamma PGA treated with the bacterial culture. Specifically, this bacteria, Bacillus subtilis bacteria, is the Bacillus subtilis Aedes 15245. So we allow to grow the bacteria for 24 hours, and we collect the material there. We collect the supernatant from there, and we applied it to this with this gamma PGA and allow to ferment, allow to incubate for another 30 minutes. Here also you can see there is no such kind of the degradation here. This 8, this is the specifically the liquid culture inhibition. Liquid culture inhibition means we add the bacteria and the bacteriophage together, and we kill the bacteria by this bacteriophages, and whatever the lysate is present in the supernatant, that one we collect it and we add this with this gamma PGA. And here you can see this is completely degradation of this gamma PGA. Another question may arise, how do you confirm this, that the bacterial maybe this gamma PGA hydrolase enzyme is produced by the bacteria itself, and it is the endophytes enzymes, endo enzymes. So that's why what we did in this 9 lane, we completely lyse the bacteria by the sonication process and collect the intercellular material, and we incubated with this gamma PGA, and here you can find there is no such kind of the degradation is present. So it is likely that this bacteriophage has some role in this gamma PGA degradation. So that's why what we did the whole genome sequencing of this bacteriophage BSP10 or the BSP18. This is a specifically, already I shown that one, this is a specifically myvirus like phages. Here you can see this genome contains the polygamma glutamate hydrolase gene. This gene is specifically responsible for the degradation of the gamma PGA. So now the question is, why this bacteriophage need to produce this kind of the enzymes? You think about that. Suppose this is the bacterial surface. This is the bacterial surface. So there are several receptor molecules in the surface of the bacteria. So what the bacteriophage do? Bacteriophage at first recognize the receptor molecule present in the bacteria. Then only it can infect, or then only it can inject its DNA to the bacterial body. So if this surface, if this receptor molecule is completely blocked by some kind of the materials like this gamma PGA, definitely the bacteriophage cannot recognize the receptor molecule. So this is the bacterial evolution. Bacteria try to protect itself from the bacteriophage attack. That's why it produced the gamma PGA. Bacteriophage is also clever. So what he did, he just produced this polygamma glutamate hydrolase enzymes. That's why this polygamma glutamate hydrolase degrade this gamma PGA, and they open up the receptor molecule present in the bacterial surface. That's why it initiates the infection process. This is the basic reason. Basically, this is a new species under the Nituna virus genus. This paper has been published in the 2018 in Viruses Journal. Next one is the BSP7. It is specifically a SIPO virus. This one is also a new genus, and it contains the homologue of the PQ0. Here you can see pure deep QE-like protein, QED, QEC-like protein. These actually help into the biosynthesis of the quinosin and arachinosis, which might help in the codon recognition. Next is the BSP4 genome. This one is also a new species under the genus Salus virus. So I am not going to in these details. So then what we did the blast in comparison of all of these isolated bacteriophages, and here we found there is at least 15 different types of the species are present. So first of all, definitely these bacteriophages are diverse. We can confirm it. I am not going in details there. So then, as these myovirus-like phages are too much prevalent in the soybean-based fermented food, so we want to find out the signature genes for these myovirus-like phages, as we can easily detect, as we can easily design the primer to detect the bacteriophages contamination in the fermenter. So we did the family order analysis, and we found it here. There are the two proteins. One is the FAM657, this is the hypothetical protein. Another one is the FAM719, which is the minor capsid protein. So these two proteins are specifically conserved throughout the Bacillus subtilis phages. So for that, here you can see there is around 88 phages of the Bacillus infecting phase, not the Bacillus subtilis. There are Bacillus ceras phase, Bacillus megatorium phase, Bacillus pumilus phase. A lot of phages are there, but these are specifically the Bacillus subtilis phase, and here you can see this hypothetical protein is conserved in this region only, and here also this minor capsid protein, it is also the conserved. So we can easily design the primer from this gene, and we can easily detect it in the fermenter or the fermentation process. So what is the conclusion? Basically, the high prevalence of the Bacillus subtilis phages in the Korean soybean-based fermented food. The infection of these Bacillus subtilis phages, BSP10 or BSP18, significantly hampered the bacterial growth in the soybean fermentation and yielded the poor quality of the product by degrading the viscous polymer. At least 15 different types of the species we identified in those Korean soybean-based fermented food, and among them, the most prevalent genus was the NIT1 virus. And we found the minor capsid protein and the hypothetical protein can be the signature genes for those myovirus like Bacillus subtilis infecting phase. Clearly, the phase analysis and the critical control point should be defined properly. So I would like to first acknowledge our honorable director of this college and principal of this college for providing me the facility to do the research in this college, and also Professor Kang Pyo Kim, under whom I did this work. And I like to acknowledge ACRB and DBT. I got the fund from them, and that's why, and also I got the fund from the college also. So that's why my lab is running. And NRF South Korea, this is the funded project actually. So I got the fellowship from this NRF, and all of the lab members here and in South Korea, and also I like to acknowledge my students, my colleagues, everyone. Thank you very much.
Thank you very much for your brilliant work as well as brilliant good presentation. Now I have got some small questions.
Yes, sir.
In India, soya sauce is being used frequently by for cooking Chinese food.
Exactly.
So soya sauce is a fermented soybean.
Soya sauce is the fermented soybean product.
Second thing, why are fermented soya foods so much prevalent or so many in number in South Korea in comparison to
Because
other?
the soybeans is the the indigenous fermented food is always prepared by the locally available raw material. Like here, you can find the milk is everywhere. So that's why we are preparing the curd in this West Bengal. So for their cases, the soybean is very much prevalent. Their productivity is high.
One more thing, these fermented soya foods are being prepared by petty shopkeepers or petty sellers or
Exactly.
petty manufacturers. Where from do they get this Bacillus subtilis? They are not scientists.
Okay, no, they are not scientists.
They are not using pure culture.
Exactly. But they are the probiotic or this Bacillus subtilis sachet is sold into the market. like us. Now you can find the yeast in the market.
Lactobacillus.
Lactobacillus, not that's the Bacillus subtilis. Yes.
Like that it is being sold
Yes, yes.
in the open market.
Open
in
market.
South Korea.
Yes, it's a packet. It's completely packet.
Is there any question from the students or researchers? Okay.
হ্যাঁ এখন পাঠান।
Thank you.
Okay, thank you very much.
I invite the
Shall I start? Shall I start, sir?
Did I ask you to
No, sir.
introduce yourself in 1 minute?
Very good afternoon to one and all present here. My name is Sudipto Mangal, Assistant Professor, Jharkhand Rai University. I have completed my M.Pharm in Pharmacognosy from Central University of Sikkim, and I have completed my Bachelor in Pharmacy from JIS University, Calcutta. My area of expertise in research is on basically on marine pharmacognosy and inland aquacultures from extraction of biomolecules from this marine aquaculture and the inland fisheries. Thank you.
So your time starts now. Start.
So the today's topic is about in vitro screening of Bellamia bengalensis for anti-cancer potential, a step towards natural cancer therapies. We know in nowadays, cancer prevalence, its incidence and its related morbidity and mortality is increasing, developing in the world, and cancer imposes a significant burden in terms of both economic cost and human sufferings. In 2024, recent statistics given that there was estimated 19.3 million new cancer cases and about 10 million cancer deaths globally, out of which the statistics given in the previous line, India ranked third after China and the United States of America. The estimated number of incident cases of cancer in India was found to be about 14 lakhs approximately. And the most prevalent types of cancers are basically the lungs and breast cancers, which were the leading sites of cancers in males and females respectively. Now the statistics given by the breast cancer of India, it was an alarming rate, increasing at an alarming rate, where 1.38 million new breast cancer cases were observed every year, and about 4,58,000 deaths from breast cancers every year. One woman dies of breast cancer in every 13 minutes in India, and in every 4 minutes, an Indian woman is diagnosed with breast cancer. Now, I have given a picture of Mother India. So, if we see the map of India, 3 parts of India is surrounded by ocean, and the internal bodies are having a rich number of brackish water lakes, rivers, ponds, and various aquaculture production is growing throughout every year. A statistics given by Food and Aquaculture Organization has given a production in million metric tons. The blue line indicates the inland production every year, which was approximately 110 million metric ton. And if we compare with the marine production, it is gradually decreasing, it is not increasing every year. So I have focused my work to do my research work on inland productions. We are all familiar with this species. Basically, this is a Bellamia bengalensis, which is found in rivers and ponds of West Bengal. And after collection of these species, I have focused on the 2 parts, basically the Bellamia bengalensis fluid and the Bellamia bengalensis mass. So, with the help of needles, I have taken this mass part and discarded these footnotes, and I have extracted my samples. This is the extraction process where I have extracted the mass and both the fluids. So, for collection of fluid, I have to prick the mass part, and with the help of needles, syringe, I have collected the fluid, and the mass was treated with PBS, and it was triturated in continuous manner. Then the sample was filtered. After doing filtration, it was gone for centrifugation. The precipitate which was formed after centrifugation was further gone for lyophilization and converted into powder form. There are some preliminary studies which I have conducted, basically hypotonicity induced hemolysis and heat induced hemolysis. So both the samples are found to be non-toxic and does not causing any hemolysis. MTT assay was performed on MCF10A cell line, which is basically a human healthy breast cell lines, and the samples does not produce any cytotoxicity effect in lower dose. In higher dose, in toxic dose, it produces approximately 10 to 15% of produces cytotoxicity. Anti-inflammatory activity was also performed on egg albumin denaturation assay and the RBC membrane stabilizations. The IC50 values which received was approximately 500 microgram per ml and 62.5 microgram per ml respectively. For this, I have conducted MTT assay on MDA-MB231 cell line and MCF7 for 24 hours. MDA-MB is basically a triple negative breast cancer cell line. So this blue line, this MTT assay was carried out for 24 hours. This blue line indicates this mollusca fluid. This blue line is mollusca fluid, and this purple line is indicating the molluscan mass. And in MCF7, this molluscan fluid is indicated in yellow color, and this red color indicates this mollusca mass. So in 24 hours, the sample showing significant activity and the IC50 value was received approximately 50 microgram per ml and 12 microgram per ml, where the fluid is showing its activity on 100 microgram per ml and 50 microgram per ml in both these cell lines. The same procedure has been conducted for 48 hours also, and the IC50 values was received on 6.25 microgram per ml for mass and 25 microgram per ml, and for fluid it is approximately about 12.5 microgram per ml and 25 microgram per ml. Staining was also performed with the help of acridine orange and ETBR. So this is the control and this is the treated cells in MCF7 cell lines, and this part was treated in MDA MB231 cell line. Here the result clearly shows the orange color that clearly shows the cells is losing its membrane integrity and causing cell death. This is all this work has been carried out in the crude part. So as per the literature survey done by me, it has been indicated that there is some sort of proteins which is responsible for its biological activity. So the primary focus of this study is to go for the compound isolation, which is basically I have done the partial purification of the purification of both the samples of mass and fluid. Here for partial purification I have gone for the ammonium sulfate cut in 4 different fractions in the percent saturation of 0 to 20%, and the percent saturation of 20-40%, percent saturation of 40-60, and the percent saturation of 60 to 80%. So in this percent saturations part, ammonium sulfate was added in different concentrations. Like in 0 to 20%, I have added 11.3% ammonium sulfate. In percent saturation of 20 to 40%, 12.1% ammonium sulfate was added, and 13%, 14%, and so on. So after adding, I have kept it in magnetic stirrer for completely solubilizing of the ammonium sulfate, then centrifuging it. After centrifugation, in percent saturation of 0 to 20%, some sort of precipitate was obtained. So in each percent saturations, all the precipitates are collected and kept for further studies. Similarly, partial purification of fluid was also done in the same procedures, and the samples are also collected for the further studies. Protein analysis was also done for all the purified fractions. In the mass part, 4 fractions we have obtained, and the crude sample is also there. So in the mass fraction third, that is the 40 to 60%, the fraction is having higher protein content, and in the fluid part, the 20 to 40% saturation is having the highest protein content. So the toughest part of the work is that all the samples has been carried out for the MTT assay to check which one is the most potent fractions, and that potent fraction is again further work for the further analysis. So the MTT assay was performed with all these 8 different samples which were obtained, which are all the different fractions, and it has shown significant activities. And this study was performed in MDMB231 cell lines. SDS page gel electrophoresis was also conducted. This part is the mass part, this is the crude, and all these are the different fractions, and this part is the fluid crude part, and all these are the fluid different fractions. So in SDS page gel electrophoresis, the mass part is having a protein content of approximately 45 kilodalton, and in fluid part the protein content is 63 kDa kilodalton and 100 kilodalton. So the conclusion of this study is the samples which was isolated from the Bilharzia bengalensis, both the fluid and the mass part, does not having any cytotoxicity activity on human breast epithelial cell line, and this result does not indicated any cytotoxic effect, suggesting and assuming that there is no harm on healthy breast cells. Protein content was estimated in both the fluid and mass fractions obtained from the mollusca, and it was essential for determining the concentration of proteins available in different crude extracts, which was important for further purification and analysis. Proteins were partially purified with the mass and extract, and it was partially purified into 4 different fractions, as I have mentioned in the previous studies. SDS-based gel electrophoresis was conducted. The mass sample revealed 45 kilodalton protein band, and the fluid sample shows band at 63 kilodalton and 100 kilodalton. Considering the 8 different samples, the most potent sample for the mass fraction is mass fraction number 3 and 4. In the previous slides, I have mentioned that the sample number 3 and 4 is having more protein content, and same result has been shown in the MTT, having better activity in MTT assay also. While the fluid sample, the potent fractions are sample number 2 and 3, also showing significant activities. These are all my references. I would like to acknowledge Dr. Pradeep Ghosh, sir, who is the founder and director of Mednipur City College, also the convener of MCCCON 2025. Thank you very much for providing me the opportunity for the Young Scientist Award and providing me the stage for valuable time. Special thanks to Dr. Shravani Pradhan ma'am, who has always supporting me, and we are also having a fruitful collaborations with Mednipur City College, providing me the premises, providing me the lab whenever I want without any hesitations. And Mrs. Ananya Datta ma'am, always supportive and helping throughout each and every research works, and all the PhD scholars and lab members of the Mednipur City College. Thank you very much. My guide, Dr. Momita Rai ma'am, presently working at JIS University, Kolkata, always motivating me, giving me the ideas.
Go to your first slide.
Sure. Sorry.
First slide.
Yeah.
First slide.
Yeah. Sure.
You know how to write in vitro?
Yeah. There is a little bit mistake. V is in
That goes with it.
V is in capital.
In vitro, is it a Greek word, Latin word, English word?
It is in vitro means outside the body, and it is basically a Greek word, I think so.
Which language?
Greek word, sir.
Greek.
Yeah.
Sorry. It's a Latin phrase.
Okay. Noted.
Understand?
Noted, sir.
In vitro cannot be written like this.
Okay.
V
Noted.
is in capital.
Yeah, yeah. This
and
is my
you have
mistake.
given hyphen of your own.
Yeah.
Anyway,
Okay. Noted,
you
sir.
have to go a long way before reaching any conclusion, before using such hi-fi title for your
Yeah.
preliminary work.
Basically, this is a pilot study, sir. So I have this is.
Let me talk.
Yeah. Sure. Sure.
Then you try to defend.
Sure.
Okay?
Sure.
I am not going to defend myself.
No, no,
You
definitely
have
not.
to defend you.
Yeah. Sure.
So, MTT test is done for what?
Basically, MTT test is run for checking the cell viability. Cell viability.
Cell wire.
Yeah.
So, by doing this MTT test, and what is the full form of MTT test?
It's a long term. It's a chemical terminology. Dimethyl tetradiazolethane 2,3,4. Dimethyl tetradiazolethane. This type.
Anyway, long term which you might
Yeah.
have forgotten.
Yeah.
by
Yeah.
this.
Yeah. Yeah.
Anyway, so by doing some cell viability test, can you claim anti-cancer potential of any substance?
Sir, I have mentioned a step
Somebody
towards.
said you have to go a long way before reaching any convincing conclusion
Right.
on anti-cancer potential of some substance.
Yeah. That's why, sir, I have mentioned.
I used to work in Cancer Research Institute.
Okay. Sir, sorry to interrupt. That's why I have mentioned a
On
step towards.
chemotherapy of leukemia.
Yeah. Sir, I have mentioned in the topic a step towards natural cancer therapies. So I have kept a step forward. It's a long step. It's a long journey.
It's you are in a preliminary stage.
Yeah, yeah, right, sir. It's in a screening stage. So further work has to be carried out.
Anyway, and which particular substance of that Bellamia prevalences do you suspect to be having this anti-cancer potential?
Yeah,
Isolated some protein fractions.
protein fractions. So the further which I have to do, I have to
You
go
have
for
to
the,
characterize those proteins.
yeah, I have to go for the ion exchange chromatography. So ion resin bands.
Your work is in its infancy still now.
Yeah, right. Right.
Okay, thank you very much.
Thank you very much,
You
sir.
have to keep up the gene.
Yeah, sure.
তাই না?
Right.
Otherwise তো কোনোদিনই এগোবে না।
Right.
এক জায়গাতেই তো start করবে। বা কিছু ব্যক্তির experience, research,
Yeah.
activities-টাই অনেকটা বেশি হয়ে গেছে তোমার তুলনায়। Thank you.
Thank you very much, sir.
The principal and vice principal, please. ওই যেটা থেকে হোক তিনজনে মিলে করবে। কারণ আমি সকাল থেকে আর একটু বেশি ওভার ওরা করছে। কারণ ওভার স্ট্রেন দিতে অনেকটা কারণ এখনো অনেকগুলো প্রেজেন্টেশন জায়গা 10টা লিস্ট দিয়েছে যার ফলে আমার পক্ষে 10টা এই আমি আমার কাগজটা বার এটা কনফিডেন্স মানে আমি আমার কাগজ তুলে নেই তোমরা কতগুলো উপরে বসবে না স্যার নিচে বসবে নিচের থেকে তোমাদের উল্টো সুবিধা হবে তোমাদের উল্টো দিকটা দেখতে পাচ্ছি একটা মিনিট আমি চাটা খাইতে
Very good afternoon. Next session is for the presentation by the research scholars. It's an oral presentation. আচ্ছা সাজিনী সিংহ রায়, বিদীপ্তা রয়, মনীষা ফৌজদার, অ্যাবসেন্ট, ক্যান্সেল, না হবে না ক্যান্সেল, না না মনীষা ডাক, পূর্ণা নন্দী, সন্দীপ দাস, পিপিকা দাস, মুকুল কুমার গিরি, মধুশ্রী প্রামাণিক, রিয়া কর, প্রিয়াঙ্কা কর, এই সরি পার্থপ্রতিম পাত্র, পার্থপ্রতিম পাত্র, মৌমিতা সামন্ত নেই, প্রণতি বেরা, অম্লান দেবনাথ, so we have 1 2 3 7 presenter. So all presenter requested to submit your PPT file to Shumanda. Mona Lisa Roy, ready. So all of you have the 7 minutes presentation. Maximum 6 minutes we will give a indications and 7 minutes stop. So basically focus on your just the background of the work, aims and objectives, result discussion, and your conclusion. So all the best. Ready?
এটা কিভাবে রাখ Good afternoon, everybody. Myself, Mona Lisa Roy. Today, I am going to present work on development of gluten-free reduced fat muffins. First of all, muffins is a very popular product. It is liked by all age group of peoples. There are several types of muffins present using chocolate chips, cinnamon, banana, various types. Muffins is generally enjoyed for as a snacks food liked by all age group peoples. Muffins are generally made of refined wheat flour, sugar and fat. But we know that refined wheat flour contains very less amount of dietary fiber and very less amount of protein, fat, etc. And sometimes the patient who have suffering from celiac disease, they cannot have it. So in this work, we are trying to replace the refined wheat flour by quinoa flour, which is a gluten-free product, and it is also known as pseudo cereals. There are several research on quinoa developed food products, so we are using this flour. The objectives of this work to make the muffin gluten-free and make the gluten-free muffin completely fat reduced. Here are some review of literature from where we have get several kinds of data regarding quinoa flour, regarding it is a nutritional value of quinoa flour, from where we can clearly see that the fat percentage is very less and the protein concentration is 13.11 gram and the carbohydrate is 53.65 gram. So from this table we can clearly understand that the quinoa flour, which is a pseudo cereal, is nutritionally very rich. Health benefits of quinoa, that is anti-obesity activity, antifungal activity, and antioxidant activity, and various defensive roles quinoa flour has. It can prevent Alzheimer's disease, diabetes, and many types of cardiovascular aging issues. There are several food products which are based on leaves, based on quinoa grains. And the second part of my research is replace the fat amounts. We have used papaya and curd as a fat replacers. Papaya and curd papaya is a carbohydrate-based fat replacer, and curd is a protein-based fat replacer. So we have used it in our product in several ratios. There is some information about papaya and curd, and the material in materials method we have presented some raw materials, that is quinoa grains, refined wheat flour, that all are bought from our local market. And the first experimental design of our product is control. In control we have used 100 refined wheat flour. Completely it is made of refined wheat flour, and the other ingredients are clearly you can observe from the slide, and 4 types of trial has been taken. From them in trial 1, 60% refined wheat flour and 40% quinoa flour has been used, and trial 2 there is 40% refined wheat flour and 60% quinoa flour is used, and in trial 4 it is completely made up of quinoa flour. Refined wheat flour is not used. And after the quinoa flour made muffins, we have used, we have tried to replace the fat amount by blends of papaya and curd. And from them 3 types of trial has been taken. From them 25% papaya, 75% curd, 50% papaya, 55% curd, and 75% papaya and 25% curd. This is the flow chart of the making of muffins. And we have evaluated the product from the sensor evaluation, and we have given maximum importance on sensor evaluation, and we have used 9-point hedonic scale, that is very popular in terms of the evaluation of food products, and some kind of physicochemical analysis was done. In the resulted discussion, all the results are done, all the analysis was done by using SPSS software. From them, first in hedonic scale, color and appearance, we have clearly observed that in 100% quinoa flour added muffins, it gives significant changes than the control, where the alphabets are same. It means there is no significant difference, but when the alphabet C is present, it means it has a significant difference from control. And quinoa has a brownish texture, brownish color, so it gives desirable color. In flavor, it also gives desirable changes, and it gives significant difference. As you can see, the alphabet C changes from A to C. Sweetness, there is no significant difference, but it is acceptable by all the sensory panelists. In body and texture, it gives desirable changes. In
Last
overall
1 minute. 1 minute left.
Okay. In overall acceptability, it gives significant changes, and it is acceptable by all the sensory panelists. Optimization of fat replacer, in color and acceptance, it gives a good amounts. All the sensory panelists was very much excited about this product. Flavor was gives good result. Sweetness also gives good result. Body and texture also gives good result. And overall acceptability, you can see that in our final product, that is 75% papaya and 25% curd. In control, there is no significant difference. From the graph, I can clearly state that there is no significant difference in our market muffins and the developed muffins in our lab. Some kind of physicochemical analysis was done in pH and moisture. moisture percentage was increased in a cake, and as the moisture percentage increased, its shelf life increased.
Time up, madam. Thank you.
What is the take-home message from your work, actually?
Actually, sir, muffins is a very popular product, but as it contains refined wheat flour, maximum consumers can't have it. So in our lab, we are trying to replace the refined flour with quinoa flour, that the nutritional value is increased and all the persons can have it.
Okay, thank you. Any questions from anyone? Any questions?
হ্যাঁ হ্যাঁ নিশ্চয়ই স্যার।
যে এসপিএস এর যে অ্যানালিসিসগুলো করেছো না, তো সেখানে দেখবে ওই যেটা এ আর বি, মানে যেগুলো এ আছে তার মধ্যে ভ্যারিয়েশন আছে কিন্তু,
হ্যাঁ আছে স্যার,
সেটা না সেটা কিন্তু স্ট্যাটিস্টিক্যালি সিগনিফিকেন্টই ভ্যারিয়েশন আছে। তুমি যদি ভালো করে চেক করো, তুমি একটু ভালো করে গ্রাফে গেলেই বুঝতে পারো। প্রথম গ্রাফটা যেটা দেখালে তুমি যাও, বলে দিচ্ছি আমি। এই যে এটাতে দেখো, তোমার যেটা রেড আর গ্রিনের মধ্যে কিন্তু ভ্যারিয়েশন আছে বাবা, ঠিক আছে?
এই দুটোর মধ্যে
প্রথমে দুটোর সঙ্গে দুটোটা কিন্তু ভ্যারিয়েশন আছে, কিন্তু সেটা তুমি এ লিখেছো হয়তো, এবং তারপরেরও একটা দুটো আমি দেখলাম একই রকম আছে, কোথাও হয়তো তোমার এখানে একটু ভুল হয়ে গেছে, একটু চেক করো।
ওকে স্যার। আচ্ছা ওকে স্যার, থ্যাংক ইউ স্যার, থ্যাংক ইউ।
Good evening, everyone. Hello. Am I audible? Good evening, everyone. Myself, Shruti Mandal, Assistant Professor, Mukberia Gangadhar Mahavidyalaya, Purbo Mednipur. Today, I am going to present a research work from our lab, development of ready-to-cook functional millet khichdi. These are my contents. So first, we all know what khichdi is. It comes from a Sanskrit word, khicha, and khichdi mainly is a combination of rice and legumes, but not necessarily it is only from rice. It may be from other cereals also, and but the legumes is constant. Like other cereals and legumes may consist in khichdi. So it consists of dal, which is a rich source of protein, and rice, which is a carb, and vegetables are added. As we all know, khichdi is a complete meal. So the basic idea is to provide protein and carbohydrates and other minerals, vitamins and fibers in a complete meal. So what we have done here, we have replaced the rice with millets. We all know in 2023, Ministry of Food Corporation has declared millet year to promote the millet consumption, because millet is a lost cereal almost, but it is very easily available and it is very popular previous days before green revolution actually. So objectives of our project was to optimization of different level of foxtail millet, barnyard millet and little millet, in khichdi. These are very not very much known millets actually, like little millet, bajra, jowar. We all know these names, but in these kind of millets, it is not very popular all over India. Though it is popular in West Bengal, we all know the kaun chal and shama chal. These are basically not rice, but it is millets. To develop nutrient-enriched, ready-to-cook millet khichdi. Now I want to focus on this ready-to-cook millet khichdi. These days, ready-to-cook meals are very popular because of our busy life. So we had tried not only to make a khichdi with millet, but also to produce it as a ready-to-eat form. Like it will, it has a very potential market value also in future. And next, optimization of the time and temperature to develop the nutritious ready-to-cook millet khichdi. What we have done, we have first prepared the khichdi and then we have dried it to prepare the, produce it as a ready-to-cook form. So these are the raw materials, basically foxtail millet, barnyard millet and little millet along with moong dal and other various kind of vegetables and spices, cooking oils. These all over food was prepared. These are some nutritional components of millet which we can clearly see. These crude fibers and protein percentage of these millets are quite high rather than the rice. So first step is the processing of millets. We have taken these three kinds of millet, washed and cooked it for, dry roasted it for 5 minutes. Then the dry roasted millets were cooked separately, and same for the moong dal. It was weighed, washed and roasted for 10 minutes and then it was ready for cooking. This is the regular preparation step which we follow for the khichdi preparation, like rice khichdi preparation. The same step is followed for millet khichdi preparation. And after the khichdi preparation, we had dried it, like tray dried it. It was, okay. It was tray dried. Various time temperature combination was experimented, but 75 degree for 4.3 hours was optimized. So these are the trials taken. As we can see, the control product has parboiled rice only, and these two have different ratios of
last 1 minute.
1 minute, okay. So different ratios of millets were taken in trial 2, trial 3 and trial 4 steps. Several sensory and proximate analyses were done, and these are the results which we can see. This 40 and 30, 15, 15, that is 2:1:1 foxtail, little and barnyard millet was optimized. So this was very much similar to the control product in every aspects. So these are some future scopes which was not yet done, but we are planning to do these all stages. Like I have already mentioned, tray drying was done.
Time up, madam.
Okay.
Thank you.
Thank you.
Next, Ria.
Question? Any question, please?
কারো কোনো কোশ্চেন আছে? আচ্ছা,
What's the new in your research actually? This
As
is
I-
traditional, people know. Millet khichdi, samachalar khichdi, or sabur khichdi, people everything know. Every people
But
knows it. But what
this
is the new?
combination is not very popular, and first of all, and it was very much similar to rice khichdi, which is already very popular. And the second thing is, it is made ready to cook form, so we can package and it is
yeah.
ready for sale.
Yeah, that's good. Thank you. Thank you. You're very good presentation.
পরপর আসি না আমরা, মানে একটা নিচ থেকে শুরু করেছি। বাড়ি কোথায়? পরপর হোক, মানে পেছন থেকে শুরু করেছি, পেছন থেকে শুরু করেছি। বাড়ি কোথায় বাবা? মেদিনীপুরেই তো। তাহলে আবার কি? রিয়া কর। কোথায় পেলি গেল? এক্ষুনি তো ছিল। আচ্ছা, মধুশ্রী প্রামাণিক। হ্যাঁ, ক্যান্সেল। মুকুল, মুকুল কুমার গিরি। কি নাম গো তোমার? ম্যাডাম নাম? আচ্ছা, ওকে।
Good afternoon, everyone. I am Sajani Singhorai. I pass out my MSc in Food and Nutrition from Calcutta University, and now I am doing research in Vidyasagar University under Professor Devidas Ghosh sir. Today, I am going to present this topic, that is diabetes management efficacy of hydroethanol extract of Bahunia acuminata bark extract in male albino rats, a preclinical approach. So we know in this present scenario we go with industrialization, urbanization and social development that causes environmental degradation and lifestyle changes, and that cause different lifestyle related chronic disorders, and diabetes mellitus is one of such disorder. Diabetes mellitus generally two types, type 1 and type 2. It has several symptoms we know, and different these factors are associated with. The major complications of diabetes are diabetic ketoacidosis, microangiopathy, microvascular complications, etc. So this is the prevalence of diabetes that is given by IDF Atlas, that shows that diabetes with people is increasing in number from 2000 to 2021, and the probability of increasing is more in 2045. So how to manage diabetes? Just follow the track. It is an abbreviation. T for take your medication at right time, R for reach your healthy weight, A for add more physical activity, C for control your HbA1c, blood pressure and cholesterol level, and K for kick the smoking habit. So there are different diabetic medications are available in the market, like insulin injections and oral hypoglycemic drugs, but in chronic use, so we choose a random plant named Bahunia acuminata, which local name is Kanchan, Plantae kingdom and Fabaceae family. So the justification of work is why we choose this plant. That is the plant has contains some phytochemicals. This plant has some folk reputation also, and it has preliminary anti-diabetic effect shown by previous in slide Sebastian et al. And it is as we use the bark part, it is very easy to collect. So the aim of the study is to search out the anti-diabetic and antioxidative effects of the extract of Bahunia acuminata bark. And before we starting the study, we take two hypotheses, that the extract may improve the carbohydrate metabolism or may it improve the oxidative status. So this is my overall extraction procedure seen here, and this is my experimental design. After acclimatization of the rats, they are divided into three groups, and diabetes control, diabetes and diabetes treated. Diabetes and diabetes treated group are given STZ injection and developed diabetes within 7 days. Then treated the extract in three different doses, 2.5, 5 and 10 mg, then treated 28 days and sacrificed, assess the biochemical parameters. This is my result. As we all know, in diabetic condition, pancreatic beta cell is distracted, so serum insulin level is reduced and fasting blood glucose level is dramatically increased. But after treatment, it has been seen that the insulin level is recovered significantly like control group, and FBG level is also improved. And here 5 mg dose treated group is the most potent group. After measuring those parameters, we measure the key enzymes, anti-diabetic key enzymes, hexokinase, glucose 6-phosphate dehydrogenase and glucose 6-phosphatase. In diabetes, due to decrease of glycolysis and gluconeogenesis, the activity of hexokinase and glucose 6-phosphate dehydrogenase in the 6 organ tissue is significantly decreased. But after treatment, it is seen that this enzyme recovery is noticed. And 5 mg dose treated group is the most potent group seen here. And in case of glucose 6-phosphatase, the level of the activity of this enzyme is increased in diabetes corresponding to the control group, but after treatment this is also recovered, and 5 mg dose treated group is the most efficient. After that, we measure the anti-diabetic key enzymes, catalase, SOD and conjugated diene. We know in diabetes, ROS production is more and water excretion is more. So the SOD catalase activity is reduced and conjugated diene is increased. But after treatment, it has been seen that the effect is more in 5 mg hydroethanol extract treated group. This is the genetic expression. We measure hexokinase and GLUT4 to notify how the biochemical parameter is good or bad. Then we measure the activity of acid phosphatase and alkaline phosphatase in ceramic level, and there is no significant difference between the treated groups. This is my histology section. In pancreatic islete, we know in control the islete diameter is more, but in diabetes the islete diameter is reduced, but in extract treated groups the islets is recovered. In case of liver tissue also, in diabetic groups the fatty plaque deposition is seen, but in treated groups the fatty plaque deposition is recovered more. This is my phytochemical screening. Qualitatively, we see the extract contain flavonoid, alkaloids and phenol, and then we go for LCMS study and we get this 9 molecules.
Done. Time up. Thank you.
What is the conclusion of your work?
Conclusion, sir, আমি যেহেতু 3টা ডোজ নিয়ে কাজ করেছি,
English, you can speak.
we take 3 dose only, hydroethanol extract in 3 dose, and we get the result that 5 mg dose treated group is the most efficient and potent in this study. So the 5 mg dose is always good in all these anti-diabetic parameters and antioxidant parameters.
Any questions?
Nice presentation and nice work. One question is that what was the group, per group, how many numbers of the animals were there?
6
First,
numbers.
and
N
second is the weight and age of the animal.
Age of the animals is at 3 months of age, and weight due to STZ injection they have to be weighted about 110 to 120.
No, is there any significant difference among the different animals in a single group?
No, sir, more or less
How
plus
much?
minus 5 or 10.
5 minus 10, 5 or
5
10
or
what?
110 plus minus 5
What unit?
gram.
Gram. Okay, thank you. Next. তুমি যাবে নাকি বিদীপ্তা? এবার থেকে শুরু করে দিই। বিদীপ্তা রয়।
Good afternoon, everyone. Myself Biripta Roy. Here I am presenting on assessment of ectoparasitic infestations in ornamental fishes of West Bengal and therapeutic potential of Calotropis gigantea extract, which is commonly known as Akondo in Bengali, under supervision of Dr. Manjit Paul, Department of Biological Sciences, Midnapur City College. As we all know, ornamental fish keeping is one of the most popular hobbies in the world. But every year, farmer faces huge losses of money all over the world for ornamental fish diseases. Diseased fish cannot be exported or marketed, and many times diseases are considered difficult to prevent and treat. Parasites are reported to be the second after the bacterial diseases in economic importance. Ornamental fishes are broadly classified into marine water and freshwater species. There are about 700 indigenous and 300 exotic marine water fish species. Indian ornamental fish has great demand in international markets, and domestic ornamental fish trade is about 500 crore, which is reported by MPEDA, and India's share to the global ornamental fish export is 0.4%, and India's rank is 31st in exporting countries. Export trade is 8.40 crore with 11.6% per year growth, which is also reported by MPEDA. And the culture of freshwater ornamental fishes is mainly limited to the states of West Bengal, Tamil Nadu, Kerala, Maharashtra, and recently in Karnataka also. Here are the objectives of the study. Firstly, to identify and characterize the prevalent ectoparasitic affecting the ornamental fishes of Howrah, North and South 24 Pargana districts of West Bengal. Secondly, to evaluate the efficacy of Calotropis gigantea extract against the identified ectoparasites under controlled laboratory condition. Thirdly, to assess the therapeutic potential of Calotropis gigantea extract in controlling ectoparasitic infestation in ornamental fishes in farm condition. And lastly, to determine the optimal dosage and safety of Calotropis gigantea extract in improving fish health and survival rates. In this part, I have collected the fishes from diverse sources of West Bengal and then examined the gill, skin and fins of the fishes under microscope. Here is the evaluation of the following parasitic indices. Calotropis gigantea is commonly known as giant milkweed, is a medicinal plant which have bioactive compounds like alkaloids, flavonoids, saponins, tannins, and antimicrobial properties also. I have used the Calotropis gigantea leaf extract here, and I have used 5 tanks marked as X0, X1, X2, X3 and X4, and X0 is marked as control tank. And 10 individual specimens were released to each tank, and antiparasite efficacy of each treatment was calculated using following equation: B minus T divided by B into 100, where is the B is the mean survival parasites of control condition and T is the mean survival parasites of the treatment condition. I have used 87 individuals of goldfishes here, and common ectoparasites under ciliate protozoa named Trichodina sp and Cynodorella sp, and Dactylococcus sp and Gyrodactylus sp under monogenians, and crustacean parasite named as Argulus sp were found. Here are some photographs of the parasites found during the study. And the graph shows that Trichodina sp has the highest occurrence and crustacean parasites were less common, and only 6 Argulus species were found yet. Okay. And the result shows that the bath treatment with 20 ml per liter with Calotropis gigantea leaf extract can remove parasites from Cariaceous aeratus after 15 days bath treatment, and much more effective than other doses comparing with controlled condition. The mortality rate was also noted, and the highest number of deaths occurring in the control group, and the highest doses, which is 20 ml per liter, was the most effective in reducing parasites intensity. In conclusion, the findings of the study contribute to the growing body of the knowledge on parasitic infestations in ornamental fishes and emphasize the importance of preventive measures in aquarium fish health management. Future studies may compare the efficacy of the extract with conventional antiparasitic treatments to establish its relative effectiveness and safety. Large-scale trials in commercial aquaculture settings would validate the practical applications of the treatment in the future. So we have discussed the findings. Here is the future prospects. Advanced diagnosis techniques implementing molecular and immunological diagnostic tools for early and accurate detection of ectoparasitic infections in aquaculture. Gill histology can be done also in fishes with parasitic infestation and after treating them with phyto extract. Combining biological, chemical and environmental control measures to effectively manage parasite infestation while maintaining fish health and ecosystem balance. So I would like to thank you.
Thank you.
So you got so many parasites from the samples, I think. So what is the basic role of this parasite in your work?
Sir, nowadays we all know that parasites are a huge reason for the losses in ornamental fish industry. So our aim is to mainly focusing on the herbal or eco-friendly treatment of ectoparasites and for environmental impact and human impact also.
Okay, nice, good one.
Thank you.
Okay, sir. Okay, sir. Good afternoon, all the respected teachers and students present here. I am Purna Nandi from Raja Narendra Lal Khan Women's College, research scholar, under the supervision of Mrinal Kanti Poida and Dilip Kumar Nandi. Today I will present a part of my research work, in vitro and in vivo experimental evidences disclose the nutraceutical effect of Ganoderma lucidum. So modern lifestyle made people more involved in medicine. It's like a cycle. We get involved in danger disease, then we taking medicine. Then we again taking the, we fall into more dangerous diseases and we again going to the medicine. So now solving to this problem, let's follow the Hippocrates. As he said that let food be your medicine and the medicine be your food. So instead of rely on any pharmaceuticals, there are many natural products are present in nature, like different herbs, different seeds, different plant, different bark extract, different leaf extract, also the different medicines. So one of the medicinal mushroom is Ganoderma lucidum. It is called medicinal because in a one mushroom there are a lot of flavonoids, bioactive components. So the main aim of this study is to investigate the role of this Ganoderma lucidum as a herbal remedy to maintain a healthy lifestyle by altering the gut microbiome, which also affect in many physiological indices. And to achieve this aim, we first, the objectives are such as to observe the changes in blood parameters before and after supplementation, also check the efficacy of this Ganoderma lucidum in some biochemical parameters, and screening the gut microbiome after the supplementation and observe the changes before and after. Also, the morphological study of intestine was observed in this study after the supplementation. So for this study, we first gather 150 19 to 21 years college going students, then taking their concern and divided into three groups. each group contained 50 students, control group which only consuming placebo capsule, GL group 1 which supplemented with the 500 mg of Ganoderma lucidum capsule for 30 days, and GL group 2 which consumed 1000 mg of Ganoderma lucidum capsule for 30 days. And then after that we starting the supplementation at the 15th and 30th day, we taking some physiological parameters and statistically analyzes all these parameters. So at the observation level, before jumping into the any physiological observation of any physiological changes, we first check the bioavailability of this Ganoderma lucidum components in human blood serum. With the help of HPLC analysis, we found that figure represent that human blood serum in which there is a major peak 3.0 096, which is close similar to the standard gallic acid 3.095, and again in the human blood serum there is a minor peak 4.046, which is also close similar to the standard epigenin peak 4.039. So this result reflecting that there is a presence of gallic acid and epigenin in the human blood serum. In the anthropometric study, there is no such significant differences we found after the supplementation comparing to the control. Weight is reduced but not significantly reduced. In the hematological analysis, we found that the WBC and hemoglobin both are significantly increased in the both supplemented group 500 and 1000 mg for 30 days, and lymphocyte and RBC, lymphocyte and platelet both are significantly increased in the second group after 30 days of supplementation, but there is no significant differences in WBC and neutrophil, MCV, MCH and HCT. In the apoptotic study, we found that after supplementation, the live cell concentration is increased significantly comparing to the control. At the same time, the apoptotic cell, early apoptotic cell and necrotic cells concentration is lower than the control in supplemented group. In the biochemical parameters, we found that there is a decreased level of glucose we found in the supplemented group for the 30 days of supplementation, and there is no change in urea, but creatinine and uric acid also shows a significant lower level.
Last 1 minute.
Okay, sir. And also we found in enzymatic assay the SGOT, SGPT also significantly decreased than the control group. In the bacteriological assay, we found that E. coli, Salmonella and Totalerus, which are mainly known as the pathogenic bacteria, significantly decreased after the supplementation for the 30 days, and as well as the Lactobacillus and Bifidobacterium, which are known as the beneficial bacteria, significantly increased after the supplementation. These are some media selective media which I have used for this study. And also the bacteria from the bacterial sample, we isolate the DNA and amplify it with the helping of universal primer following the PCR method. In the cell viability assay, we check that the triple negative cancer cell line control cell lines are showing cell viability higher in the comparing with the. Okay, sir. But
Would you show your title of the work?
Okay,
In vitro and in vivo experimental evidences disclose the nutraceutical effect of Ganoderma lucidum.
Okay.
It is the food supplement. It is approved.
Yes, sir. It is approved by Ayush.
Ayush.
Yes, sir.
West Bengal.
Huh.
Ayush India.
Yes.
Actually, you have you any approval for Human Ethical Committee?
Yes, sir.
Could you please show?
Huh. This is the human ethical. Also, it is the animal ethical because.
No need of animal ethical. You did not check anything in animal.
Sir, actually, I have not finished. Actually, I have a same study also.
You have to start your work from animal, then
No.
cell, animal, then human. This is the way of starting. This is not the way for starting. Okay?
Okay.
Another one, you have checked different blood parameters.
Yes, sir.
Go. Blood parameters. What you want to check actually?
Sir, we have to check the blood total CVC.
Why CBC? What effect you want from Ganoderma?
Because we from the literature review we found that it can increase the energy in animal model, so we want to found in human as well as that is it preferable for increasing the energy hemoglobin concentration for females and RBC, and also it can be used for the anemic condition. If it increase the RBC level or hemoglobin level, that it could be a preventable method
If
for.
hemoglobin is too high, then she is healthy.
Yes, sir.
If hemoglobin is too high,
No, sir.
then she is
Too high.
healthy.
No. Too high is not healthy, sir.
And why you have checked the bacterial plaque different?
Bacterial.
Why?
Sir, this
From
mushroom.
which sample?
Human fecal sample.
Human?
Human fecal sample. Stool sample.
Stool sample. Okay. Thank you.
Okay, sir. Thank you.
Hello. Contact. It next. Hello. Good afternoon. I am Mukul Kumar Giri, research scholar, Vidyasagar University. My topic is amelioration of pyrethroid-induced male reproductive toxicity by probiotic-rich nutritional supplement formulation in albino rat. Pyrethroid have been identified as endocrine disrupting chemical which are responsible for the male reproductive impairment. Some epidemiological studies have reported environmental exposure to pyrethroid may alter the reproductive health of adult male subject, mainly in terms of reduce the sperm concentration, quality, and damaged sperm. So, in this study, we have designed a probiotic-enriched nutritional supplement formulation for the management of pyrethroid-induced male reproductive toxicity. It is my aim and objectives to search out the pyrethroid-induced reproductive toxicity in male albino rat. Second, to find out the protective efficacy of formulated probiotic-rich nutritional supplement on pyrethroid-induced complication by biochemical, molecular, and histological technique. It is my experimental design. 18 rats are divided into the 3 groups. Each group contains the 6 rats. First control group received only the vehicle. Pyrethroid groups treated with the 10 mg per kg body weight. Then pyrethroid plus supplement group. These groups are treated by pyrethroid 10 mg per kg body weight. After this treatment, supplements are provided to this group. Supplement means it is the mixture of the probiotics from the human milk and extract of pumpkin seed, 3 ml per kg body weight. After 45 days of the treatment, rats were anesthetized for collection of the blood and organs and then euthanized. Blood and organs was collected for the biochemical, molecular, and histological study, then statistical analysis of the data. It is the methodology part. It is my result. The final body weight is significantly reduced in pyrethroid group in compared to the control group, but in supplement group means the mixture of the probiotics of the human milk and the extract of the pumpkin seeds. It is significantly restored towards the control level. Table 2, it is the organosomatic index. Organosomatic index of the testis, epididymis, seminal vesicle, prostate is significantly decreased in the pyrethroid group, but in supplement group it is recovered towards the control level. It is the sperm count, sperm motility, viability and Hos level is significantly decreased in the pyrethroid group in compared to the control group. But in the supplement group, it is significantly recovered towards the control level. Second, testicular cholesterol, seminal vesicular fructose level is high in pyrethroid group in compared to the control group, but in the supplement group, it is recovered towards the control level. The delta 5 3 beta HSD, 17 beta HSD is decreased in the pyrethroid group, but in the supplement group it is restored towards the control level. The CD, MDA level, SGOT, SGPT is comparatively high than the control group in the pyrethroid group, but in the supplement group it is significantly recovered towards the control group. It is the serum testosterone level, LH, FSH, SOD, CAT, GPX level is significantly decreased in the pyrethroid group. Pyrethroid means the one type of the pesticide that destroy the testicular tissue. But in the supplement group means the mixture of the human milk probiotics and the pumpkin seed extract. This group, this enzyme and this hormone are significantly recovered towards the control level. The band density of the antioxidant enzyme SOD, CAT, GPX in the pyrethroid group or the B group is reduced in compared to the control group, but in supplement group this antioxidant enzyme level gene expression is comparatively better after the treatment of
last
supplement.
1 minute. 1 minute left.
It is the histoarchitecture testis at magnification of the 40x, control group showing the normal stage of spermatogenesis with the normal cell density, pyrethroid group showing the degeneration of the germ cell, but in the supplement group increase the number of the germ cell and spermatid towards the control level. It is the lactobacillus bacterial colony. The CFU, colony forming unit in the pyrethroid group is low compared to the control group, but after the supplementation the colony forming unit in the intestine is increased. It is a discussion, sperm profile, testicular cholesterol, seminal vesicular fructose level, GOT, GPT were elevated, but serum testosterone, FSH, LH level were decreased significantly in the pyrethroid group in respect to the control group. Conclusion from the above study, we may conclude that mixture from of probiotics of human milk and pumpkin seed extract 2:1 ratio has a defensive role for the restoration of pyrethroid induced male reproductive damages.
Time up. Okay. Just one word or sorry, one line. Can you explain that what actually you wanted to do, what you obtained, and what is your findings?
Actually, infertility is the nowadays it is a global problem. Probiotics and pumpkin seed extract. Probiotics and pumpkin seed extract increase the sperm profile, means the sperm motility, sperm morphology. So I study the pilot research. Human milk probiotics is the better, gives the better result compared to the any types of the probiotics from idli and curd.
Okay, thank you.
Good afternoon, respected delegates member present here. I, Madhusri Pramanik, presenting on behalf of my co-authors, my work entitled Eugenol Oleate Pursued the Immune Response Against Drugs-Resistant Leishmaniasis Donovani under the supervision of Dr. Shantanu Kaur Mohapatra. As we all know that leishmaniasis is a neglected tropical disease caused by the protozoan parasite Leishmania and transmitted by the phlebotomine sandfly. Leishmaniasis is mainly divided into three groups. All the leishmaniasis is mainly visceral leishmaniasis ineffective in Indian subcontinent, and all have the same type of symptoms except the infection site. At first, the Leishmania donovani forms the intracellular promastigote, which transform into extracellular amastigotes, which leading to the severe immune suppression, leading to visceral leishmaniasis. If we talking about the epidemiological part, mainly in Indian subcontinent, visceral leishmaniasis is effective, and mainly in India, Bihar, Uttar Pradesh, Jharkhand, and West Bengal have the many cases of visceral leishmaniasis. Here are some treatment options for visceral leishmaniasis, but the major problem is drug resistance, so we need the urgent for better treatment option. Among the derivatives of eugenol, we choose the eugenol oleate as it has the antipromastigote and anti-amastigote activity with the high therapeutic index, which induces the antiviral immune responses in murine macrophages and bulbs mice, which activated the PKC-P13 MAPK pathway to induce the anti-leishmania immune responses. Extensive use of anti-leishmanial drug in subtherapeutic doses, incomplete duration of treatment, and substandard drugs enforce the drug resistance in 60% of VL, which is a serious issue in India. Nowadays, stibogluconate is not effective. Amphotericin B and miltefosine became clinically resistant, while the permomycin is not recommended for VL. So, recent report indicate that the drug-resistant VL strain does not affect on immune modulation. So, we can hypothesize that the eugenol oleate have proved itself as a potent antiviral molecule and possesses efficacy through immune modulation. Hence, eugenol oleate may cure the drug-resistant VL through activation of host immune responses. The aim of my work is to establish the potential of eugenol oleate against drug-resistant strain of visceral leishmaniasis. To fulfill this aim, here are two objectives. First of all, to study the anti-leishmanial effect of eugenol oleate against drug-resistant strain, and to examine the immunomodulatory properties of eugenol oleate during drug-resistant visceral leishmaniasis. For this work, we choose the 4 strains of Leishmania donovani, like sensitive strain AG83 and other the resistant strain like amphotericin B resistant, SAG resistant, and permomycin resistant. We use bulb C mice for in vivo and in vitro work. As we come to the result and discussion part, at first we synthesize the eugenol oleate as it is not available in the market. We purified the eugenol oleate by column chromatography method and characterized by proton NMR and carbon NMR. Then we infected the macrophages by 4 strain of Leishmania donovani strain, and after 48 hours we treated with 5 microgram per ml of eugenol oleate, and we can see that the maximum 80% decrease of parasite load by using the gym sustain. And we also check the NO generation, which is the effector molecule, to check the parasite burden, that the maximum 20-fold increase in AG83 strain and more than 10% fold change appeared in the resistant strain. We also check the cytokines value like pro-inflammatory like INOS2, IL-12, IFN gamma, where we can see that the AG83 when we treated with the eugenol oleate, we can see that the parameter increase, and when and in anti-inflammatory cytokines the parameters are decreased with the eugenol oleate. Here we can see that the pro-inflammatory increase, and it has an indication that the INOS is effective on this parasite burden. After that we get a maximum good value in vitro result, so we go for the in vivo experiment. Here we choose the 7 group, like first of all uninfected control, AG83 infection plus treatment group, amphotericin B
Last
resistant,
1 minute.
amphotericin B resistant infection, and the other SAG resistant infection. At first we check the hepatic LDU and splenic LDU, where we maximally see that the 80% parasite burden decrease in this with this treatment. T cell, we also check the T cell proliferation, which is the indicator of Th1 and Th2 proliferation, where we can see that the T cell proliferation increase with the treatment group, and also the nitric generation is also increased compared to the control group. Here we can see that the Th1 and Th2 cytokines in infected biopsy mice, where we can see that the IL-12, IFN gamma, IL-10, IL-12, IFN gamma and INOS expression is increased, and IL-10 and arginase 1 expression in decrease. So we can conclude that when we treated with the eugenol oleate, the pro-inflammatory is...
Time up. Say thank you.
Thank you.
What is the conclusion of your work?
Sir, conclusion, যখন আমরা eugenol oleate
In English.
Sir, when we treated the infected macrophages with eugenol oleate, we can see that the maximum increase in pro-inflammatory like INOS, and we know that when INOS expression is increased, it induces the NO generation, which may kill the parasite in macrophages.
You are showing MFI. Show me the slide. MFI. What is meant by MFI? What way you have measured the T cell proliferation? Have you any photo from where you make the MFI? From where you make the MFI? You are showing this bar diagram only.
Yes.
So, what we can believe this is true MFI? You have to show your photograph, please. Mean fluorescence intensity is MFI. You have to check by photograph. You have to show when you are showing the MFI, that time you have to show your photograph of T cell proliferation. Proliferated T cell, তুমি antibody দিয়ে দেখাও, tag করো। না, assay result হবে না। Okay. Thank you.
Thank you.
Good evening, respected professors. Good evening, respected professors and research scholar present here. I am Riya Kaur, research scholar of Midnapur City College under the guidance of Dr. Shravani Pradhan, Assistant Professor in Nutrition, Department of Paramedical and Allied Health Science, Midnapur City College. Today I am here to present a part of my research work, entitled Gondoic acid attenuates high-fat diet-induced obesity in mice by modulating the expression of key adipogenic genes and activating AMPK and GLUT4 pathway. So as you all know, the overview of obesity, well known. So the scientists stated that
obesity management via sea fish oil is a fruitful way now, and fish is regarded a budget-friendly source which contain diverse range of PUFAs and MUFAs, and it's have beneficial effect in our body. However, omega-9 fatty acids have recently received a wide attention due to its emerging effects in our body. So, in this study, for the very first time, we want to establish the anti-obesity effect of omega-9 gondoic acid, which very first time for characterization for topse fish oil. So, the main aim of our study is to establish the anti-obesity potentiality of omega-9 gondoic acid in mice model, and to fulfill this aim, here are some objectives. First, characterization of marine P. paradiseus fish oil, then to search out the anti-obesity potentiality of newly characterized gondoic acid in vivo model. This is the concerned methodology with my work plan. At first, we taken total 36 number of bulb C mice and segregated the mice into 6 group: control group, positive control group, obese control, treatment 1, treatment 2 and treatment 3 group. All the mice group contain total 6 number of mice, and all the mice fed with normal diet for first 7 to 14 days to adapting the normal environment. After that, except the control and positive control group, all the group fed with high fat diet. And after developing the obesity, it will take time 8 to 10 weeks. Treatment 1, 2 and 3 groups supplemented with gondoic acid with 3 different doses. For treatment 1, it was 1 mg per 100 gram body weight per day. For treatment 2, it was 2 mg. For treatment 3, it was 4 mg per 100 gram body weight per day. Total experiment was continued for 16 weeks, and then we analysis the lipid profiles, oil red wine HE staining for adipose tissue and liver stain, and then RNA isolation from obese cell line, and check some expression of obesity related core markers, and lastly protein analysis by western blot. Following the objective 1, here figure 1 represents the GCMS characterization of P. paradiseus fish oil, which clearly reveal the presence of 13 PUFAs and 10 MUFAs. Here all are listed along with their RT values. Among them, we chose gondoic acid. We get it for 47.87 RT value and use it for our further studies. Following objective 2, figure A represent the variation of fat accumulation in different experimental mice group. Figure B represent the variation of food intake in different experimental mice group, and figure C represent the variation of body weight, where you can see in obese control group the mice have tendency to gaining the more weight than the other groups, and figure D represent the BMI. So during the animal experiment, we also measure the length of the animal, and using the length and weight we calculate the BMI. So figure D represents the variation of BMI, where where obese control group having the higher BMI than the other groups, and after treatment of gondoic acid, it was started to decrease the weight in all the other groups. And figure EFG represent the variation of peripheral organ weight in different experimental groups. Then we check the biochemical analysis, where we see that serum glucose level, cholesterol, triglyceride, VLDL and LDL level was higher in obese control group, and after the treatment of gondoic acid, it was started to gradually decreases in all the treated group, mostly in treatment 3 group. And HDL cholesterol level was basically lower in obese control group, and it was started to gradually increase after the treatment of gondoic acid. And side by side, we also check some expression of toxicity related markers like SGOT, SGPT, serum urea and serum uric acid. All the levels were reduces in the obese control group. Then we check the DAPI staining and HE staining in liver tissue, where we see in obese control group the fat accumulation level was very high than the other groups, but after the treatment of gondoic acid, it was reduces in the treated group. And in DAPI staining, we check the liver tissue, liver morphology, where you can see in obese control group the cellular density of liver was very high, but after the treatment of gondoic acid, it became in normal cellular density in liver tissue. Then we check the fat dipolate accumulation in the white adipose tissue, where we also see in obese control group the fat accumulation level was very high, but after the treatment of gondoic acid in different doses, it was mostly reduces in treatment 3 group. Then we check some expression of obesity related marker, where you can see the core markers AMPK, adiponectin, GLUT4, CPT1, all level was higher in
Ria, last 1 minute.
Lower in treated group and higher in obese control group. And we also check some expression of inflammatory marker. We can see after the treatment of gondoic acid, the inflammatory marker level was reduced in the treatment group, and the anti-inflammatory marker was reduces in the obese control and treatment group gradually. To confirm our result, we also done protein analysis. Here we see the AMPK and OB level was higher in treatment group, and CBP, SRBP, FAS and PPR gamma level was lower in higher in obese control group. After the treatment of gondoic acid, it lower in the treatment group. So we concluded that gondoic acid is a 20 carbon monounsaturated fatty acid. We characterize it from the topse fish oil. After the treatment of gondoic acid, we following our result concluded that this fatty acid have the anti-obesity potentiality. Here we use 3 different doses of gondoic acid, and among the 3 doses, the 4 mg per 100 gram body weight per day, this is the effective dose.
Thank you, Bal.
Publications. Thank you.
First result. First result. GCMS, right? GCMS. GC or GCMS? Which one you have done? For the analysis of the oil. Is it? So, GCMS or GC? GCMS.
GCMS.
So, what was the column and
Sir,
what was
we
the column
don't
and
know
what was
because
the, no, you can't say that.
Okay, sorry, sir.
You have to answer this
Okay,
one.
okay.
What was the column and what was the mobile phase of this analysis?
The column was, I think, C15 for fatty acid. And sir, actually this result is not done here.
Anywhere it can be done. That
it
can
is
be, but
from
this information is basic and you have to write in the materials method, madam. That is
Sir,
acceptable
methyl
anywhere.
fraction used for mobile phase.
Huh?
Methyl fraction.
Methyl fraction, no. My question is for the GC of your sample, what was the column and what was the mobile phase? And it's a very general question, means anyone will ask that. Because from your analysis, you have identified the compound, right? Based on the major peak height, peak, everything. So that, I think, okay. So coming to the other part of the, another one. So, so you have induced the obesity, then you have treated the oil. Am I right?
Yes.
So what was the initial weight? Can you show any body weight data? So this is your body weight data, right?
Yes.
So what was the initial weight? What was the increase and how much you have decreased after your supplementation or treatment?
Initial weight was, sir, 18 to 20 gram, and that time my age was 4 to 5 weeks. And after the obesity development, weight was almost 50 gram. And when we started to use the fatty acid, after 6 weeks, it reduces like 35 to 40 gram. Thank you, sir.
Next, Pipika. Huh? Oh, tarpore. Latest, late fine. Chafa khawabe tarpore.
Good afternoon to my respected professors. I am Pipika Das, working as an Inspire Fellow of Biodiversity and Environmental Studies Research Center under the supervision of Dr. Shaboni Pradhan, Assistant Professor in Nutrition, Department of Paramedical and Allied Health Science. Today I am going to present a part of my research work entitled Effects of Tapra Fish, Opisthophthora staduri oil on hepatic lipogenesis and inflammation in mice with diet-induced obesity, a time-dependent mechanistic study. Basically, obesity results results from the disequilibrium of energy intake, that is food consumption, and energy expenditure, like the basal metabolic rate, that is the biochemical processes required to maintain cellular viability, physical activity, and adaptive thermogenesis. Adipogenesis is the process where pre-adipocytes mature into adipocytes accompanied by changes in morphology, hormones, and gene expression, which involves the pathological changes and accumulate in different organs, leads to visceral obesity. And considering the adverse effect of drug and the beneficial effect of polyunsaturated fatty acid present in the marine sources, the main goal of my study to develop a potential compound by using locally available marine fish. Opisthophthora taduri, commonly known as tapra fish, is a low-cost marine fish which is easily available in the Bay of Bengal region. So the objective of my study to investigate the anti-obesity potentiality of oil extracted from O. taduri on high-fat diet-induced obese mice in a duration-dependent manner, and investigated the primary molecular mechanism underlying these effects. These are the concerned methodology. At first, fish was collected from the coastal area of Purbo Medinipur district, then oil was extracted by standard solvent extraction protocol, and this is the overall in vivo experimental design. First, male albino mice, 36 male albino mice, 4 to 5 weeks, and the weight is 10 to 15 gram, was divided into two groups. First one is control group, which fed the normal diet. Second one is obese control group, which fed the lard-based high-fat diet. And following 12 weeks of high-fat diet, mice was again segregated into another 4 groups, which is co-administered with O. taduri oil at a dose of 600 mg per kg body weight for 14 days, 21 days, 28 days, and 35 days. And after the treatment period, mice was sacrificed after overnight fast, and the different biochemical, histological, and genomic analysis was performed. Visual observation revealed that mice fed with high fat diet showed the greater body weight than mice fed with the normal diet group, and O. taduri oil significantly reduced the body weight as well as the BMI, especially in the last 2 weeks of the experiment, and also reduced the BMI, especially in the last 2 weeks of the experiment. I also found a slightly altered food intake pattern, but it was statistically insignificant. Then I have checked the blood lipid profile, and the serum analysis depicted that O. taduri oil significantly reduced the level of total cholesterol, triglyceride, VLDL, LDL, and increased the level of HDL, which showed the hypocholesterolemic and hypolipidemic properties. And as the dysfunctional metabolism of glucose and lipid is directly linked to obesity driven by high fat diet, I also checked the fasting glucose level, and O. taduri oil significantly reduced the fasting glucose level with the increment of concentration of total protein in a duration dependent manner. Analysis of liver function is crucial for determining any type of metabolic disorder driven by obesity, and this result indicated that there is no increment of serum GOT, GPT, and ALP level, and previous report showed that some marine fish oil may increase the uric acid content. That is why I have also checked the urea and uric acid level, and this result indicated that there is no increment of urea and uric acid level or the renal dysfunctioning. Grossly, the livers of normal diet group showed the dark brown or red color of the liver, whereas the high fat diet were shown the yellowish and lackluster, and fish oil had recovered the dark brown and red color of the liver and also reduced the relative weight, and histologically many lipid droplets and fat vacuoles were present in the high fat diet group, which is also lowered in the last two treatment groups. And adipocyte quantity and adipose tissue weight were also measured in order to investigate whether the weight reducing effect of fish oil was due to decrease in fat mass, and this result indicated that there was significant reduction of visceral adipose tissue weight, and histological staining demonstrated that there was a significant reduction of inner diameter of adipocyte size in comparison to high fat diet group, which is more closely matched observation with the normal diet group. Then I have checked several lipolytic and anti-inflammatory gene expression by semi-quantitative PCR, and this result indicated that there is increment of LPL, AMPK, CPT1, PPR alpha, and adiponectin by induction of fatty acid beta oxidation, as well as the anti-inflammatory mRNA expression like IL-10 and IL-1RA. Then I have checked several adipogenic or lipid synthesized gene expression, and this result indicated that there is no there is decrease mRNA level of CBP alpha, BPR gamma, FASH and SREBP1C by reducing the de novo synthesis of fatty acid and fat cell differentiation. Also, the oil has the capacity to reduce the risk of leptin resistance in obese mice. Then I have checked the adipogenic or lipogenic gene expression from liver sample, and the same result was shown in this figure. And then the immunoblotting analysis also depicted that there was significant upregulation of AMPK CPT1 adiponectin protein expression and reduction of OP and FAS PPR alpha. And these are the possible molecular mechanism by which fish oil exert anti-adipogenic potentiality. In summary, the fish oil can activate AMPK, which can reduce the acetyl CoA synthesis and melanin CoA, and lower level of melanin CoA can increase the CPT1 expression. Based on the evidence from my present study, it may be concluded that over a 4 and 5 week treatment with designed to fish oil treatment followed along with high fat diet can exert anti-adipogenic potentiality by regulating the hepatic lipogenesis and adipose inflammation.
Thank you. Go.
In one result, you have shown that the color of the liver has been changed during the. This is the first observations, or is there any other references on it?
Yes, sir, references.
So, can you explain it?
Because obesity, after obesity, there was accumulation
can
of.
You show that figure?
There was accumulation of lipid droplets, and this can result the non-alcoholic fatty liver disease. But after fish oil treatment, the dark brown or red color, as because the fat droplet number and was reduced in the treatment groups, that is why the color was dark brown or red color.
Okay.
Okay, sir. Thank you.
Okay, thank you.
Good afternoon, everyone. I am Manisha Fauzdar, research scholar of Minnapur City College, working under the guidance of Dr. Shravani Pradhan, Assistant Professor, Department of Nutrition. Today, I present a part of my research work entitled Narvonic acid-induced apoptosis on MDMB 231 cancer cell line via ROS generation and apoptotic gene activation. As we know, the incidence of breast cancer are increasing day by day. As we know, the incidence of breast cancer are increasing day by day, and breast cancer is the 5th leading cause of cancer-related death among women. According to Globcan 2020 data, cancer currently one of the most frequently diagnosed malignancy is breast cancer in women, with an anticipated 2.3 million new cancer cases worldwide. By 2040, due to population growth and aging, the incidence of breast cancer is expected to rise more than 3 million per year. So there is a great urgency to develop some therapeutic strategy for prevention and treatment of breast cancer. Different previous literature already established that different PUFA and MUFA have anti-cancer potentiality. So in our study, we select a monounsaturated fatty acid, which is narvonic acid, which is a very long chain monounsaturated fatty acid. Narvonic acid has a close relationship with brain health and also plays an essential role in immune regulation and has anti-inflammatory properties. So the aim of my study is to determine the anti-cancer potentiality of narvonic acid, which will be used as an effective tool for breast cancer treatment, and the objectives are to determine the anti-cancer efficacy via ROS generation and apoptotic gene activation, and also study the apoptotic morphological changes using DAPI and acridine orange ETBR staining. This is the proposed work plan. The result figure 1 shows that the cell viability detecting by the MTT assay, as we see in this figure 1A, 2A, that after treatment with narvonic acid at different dose and duration, it decreases the cell viability of breast cancer cell line. And figure B shows the effective result, which is showed in 24 hours treatment, that after 24 hours treatment, narvonic acid reduce the cell viability in breast cancer cell line. This is the ROS generation. Figure 3A shows that after treated with narvonic acid at different doses, it increased the ROS generation within the breast cancer cell line, and figure 3B shows the quantitative measurement of ROS production within the breast cancer cell line after narvonic acid treatment. This is the figure 4, shows the different oxidative marker which increase the oxidative stress within the cell. GSH, which is an antioxidant which decrease after treatment with narvonic acid, and GSHG, nitric oxide, and MDA level are increased after treatment with narvonic acid, which is an indicator of oxidative stress or increased ROS generation within the cell. This is the different gene expression study. As we see in this figure, P53, BAX, BCL2, caspase and caspase 9, caspase 3 and caspase 9, these are the different apoptotic and anti-apoptotic gene. The apoptotic gene expression are increased after treatment with narvonic acid, and anti-apoptotic gene expression are decreased after treatment with narvonic acid. This is the acridine orange ETBR staining. As we see in this figure, after treatment with narvonic acid, the breast cancer cell death are increased. As we see in this figure, that acridine orange staining is higher in the control group than the treated group. This is the DAPI staining. As we see in this figure, after treatment with narvonic acid at different doses, the number of DAPI positive cell are decreased. And this is the tunnel staining. As we see in this figure, the tunnel positive cell are increased after treatment with narvonic acid. So we can say that after narvonic acid treatment, the cell viability is decreased. Different apoptotic gene marker are increased, and anti-apoptotic gene marker are decreased. Also, it increased the malondialdehyde and other oxidative stress marker. So we can conclude that after treatment with narvonic acid, the breast cancer cell line, in breast cancer cell line, it can be treated as a breast cancer therapeutics. So these are the references I would like to acknowledge.
Okay. What is the relevance or rationale for the choosing the mavulonic acid?
Sir, I choose narvonic acid because I mean narvonic.
Narvonic, yeah.
Yes. Sir, because it has some anti-inflammatory properties, so we choose this acid, choose this fatty acid to check that these have some anti-cancer.
You are also checking the anti-cancer activity, right?
Yes, sir.
So already that has been reported, as per your information.
And anti-cancer efficacy is not checked. Anti-inflammatory efficacy was checked before that.
Okay. So there are lots of other molecules are there which have the anti-inflammatory, anti-obesity. Even your lab is working on different compounds.
Yes, sir.
So what is the specific rationale of choosing this narvonic acid?
Because no previous study done on these fatty acids.
বলে হবে না, তোমাকে তো সায়েন্টিফিক্যালি প্রুভ করতে হবে যে কেন তুমি চুজ করেছো। কি বলছেন?
ঠিক আছে ওটা ম্যাডামের আর ওর ব্যাপার। আচ্ছা, যেটা আরেকটা কোশ্চেন, তোমরা খুব সিলি কোশ্চেন অ্যান্সার দাও তোমাকে। হোয়াট ইজ মেন্ট বাই ইনফ্ল্যামেশন? হোয়াট ইজ দ্য ডেফিনেশন অফ ইনফ্ল্যামেশন, ইমিউনোলজিক্যাল ডেফিনেশন অফ ইনফ্ল্যামেশন? আর ইনফ্ল্যামেশন ইজ গুড অর ব্যাড, অর ইট ইজ অলওয়েজ ব্যাড?
No, sir, inflammation is always not bad. Sometimes it is good, but extreme inflammation is bad.
And what is meant by inflammation? What is the definition of inflammation?
When there is increased inflammatory marker within.
Yeah, okay. When inflammatory marker is increased, then it is inflammation.
No, sir, in inflammation there is increase.
থ্যাংক ইউ, ভালো হয়েছে, তোর কাজ ভালো হয়েছে, যা।
Then we are closing the session. Thank you very much for the speaker and your patience. Thank you.